Hello everyone, good evening to all. Welcome to another class. Today we are going to study about the protein sequencing. What we are going to study about protein sequencing. Okay, so before moving further, let me just ask one thing. What is protein? What is protein? आप सब प्रोटीन से क्या समझते हैं? What do you all understand when the word come protein? Basically, let me answer. I cannot take your answer because I'm not live. This is the recorded classes. So protein is polymer of amino acid. Okay. To get what amino acids are there, to know about the amino acid, to know about the amino acid. Like if we want to know what are the acids which are in the protein, we all know that it is 20 standard amino acids which proteins made of. What which 20? What are their sequence? To know the sequence, we do the protein sequencing. What we do? We used to do protein sequencing. Okay. So first protein sequencing was basically given by Frederick Sanger. You must have heard about the word Sanger much a time when you are seeing the protein sequencing lecture or reading the books or seeing any article. Sanger is the first person who worked out for the sequence of amino acid residues, who first tried and first developed to get the sequence of amino acid. He tried on the first what? Hormone, bovine insulin. He first tried on the bovine insulin hormone. First he do the sequencing of this protein of hormone protein. He do the sequencing. Okay. So I have already told that a protein is made up of amino acid sequences. There are various different amino acids which are in the protein. Okay. So first principle was given by Sanger only. Okay. So let us see further. As you can see, this is a polypeptide chain. This is our polypeptide chain. Polypeptide chain of amino acid. We can say polypeptide chain of amino acid. Okay, so we all know that there are two terminal in a polypeptide chain. Okay, one we called N terminal. Okay, another we called C terminal, which is present at the you can say right hand side and left hand side. We have the N terminal. Okay, we can say different amino acid are sequenced here. You can say different colors of balls. It is sequenced here. Okay, and between two amino acid there is what? There is a peptide bond. There is a peptide bond. Two amino acids are joined by peptide bond. Okay, two amino acids are joined by peptide bond. Suppose one amino acid is this and one amino acid is this. So what we call this? We call this dipeptide. What we used to call this? Dipeptide. Okay. If you want to see the structure, just you assuming the structure, you can look after it. Just look. This is the amino acid structure. You can see it is zooming right now. Okay. Now you see. This is the amino acid structure. This is the amine group. Okay. This is the R chain. This is the acetic group. Okay. And the hydrogen atom. And this is the alpha carbon. Okay. This is what? Alpha carbon. Basically, our protein are majorly made up of L amino acid. It is majorly made up of L amino acid. So we need to the sequencing of this. Okay, we need the sequencing like what are the amino acid which are sequenced in this. Okay, let's move further. So first step. for the sequencing is acid hydrolysis. Like we have seen in the previous slide, there are different amino acid in a polypeptide chain. Okay, this you can see chain, polypeptide chain. Okay, you can see there are different amino acids. Okay, the different colors indicate the different amino acids. Okay, first we need to know how many amino acids are there. Like we need to understand, like suppose if we are having one chain, like this is the chain, peptide chain I can say, we need to see how many amino acids are there, like how many are alanine, how many are lysine, how many are phenylalanine, how many are tyrosine, how many are tryptophan. We first need to see the number of amino acid we had. Okay, for to know the number of amino acid, we first step was acid hydrolysis. First step was acid hydrolysis. Acid hydrolysis is the first step. Okay, so this is the first step, acid hydrolysis. To do acid hydrolysis, how we do the acid hydrolysis? We do the in the 6 molar concentration of HCl. Okay, 6 molar concentration at the temperature of 110 degrees Celsius for 24 hours. We do the acid hydrolysis. After the 24 hours, this is the 24 hours, what happened? Our amino acid gets separate. Our amino acid get separated. Okay, we keep the peptide bond. We used to the peptide bond. Okay, so our amino acid get separated. Okay, after getting these amino acids separated, how we can come to know that which amino acid is what? How we cannot see amino acid with a naked eyes. Okay, so we need to know which amino acids are the are in the polypeptide chain. Okay, to know about that, we'll come to the just a minute. Sure. We'll do the HPLC. We'll come to the HPLC. We pass our amino acid sample to the HPLC. Then HPLC, just I'll again just show you the zoom one diagram. It will be shown to you soon. Yeah, just a minute. It's coming. Okay, you can see this is the HPLC. Okay, chromatogram. You can see here the amino acid concentration for serine, for glycine, okay, for alanine, okay, for threonine, okay, you can see for proline, for tyrosine, for valine, okay, for methionine. It tells about amino acid concentration, like what amino acids are there, how many amino acids like there. Suppose I say two alanine are there, okay, four lysine are there, five phenyl phenylalanine are there, six tryptophan are there, okay, and I say three glycine are there, okay, and four tyrosine are there. So why doing the SPLC we get the number of amino acid. Okay, what we get? Number of amino acid. Just a minute, I'll show you. Sorry, just a minute. Yeah. Okay, this you can see this is the result we get from the HPLC that this is the sequence of amino acid. The like not we don't get the sequence now. We first got the number of amino acid. What we get? Number of amino acid which we have in the polypeptide chain. Like how many alanine we have, how many lysine we have, how many phenylalanine we have, how many tryptophan we have, how many glycine we have, how many tyrosine we had. By this we get the number of amino acid. Now our third step. Now our third step. Sorry, second step. It was first step. What we have done? We have done the hydrolysis. Okay, hydrolysis completed. Just a minute. Okay. Hydrolysis completed. After getting the number of amino acid by SPLC, we come to know what are the number of amino acid we have in our polypeptide chain. Okay. Then we come to the second step. We come to a second step. Second step is N terminal analysis of C-terminal analysis. We need to know which is the amino acid on the N-terminal and which is the amino acid on the C-terminal. We can know this by doing the N-terminal amino acid and C-terminal amino acid. Okay. Sanger was the first person. Sanger developed the reagent for this. Sanger was the first person who developed the reagent for this N-terminal amino acids. What he developed? 1-fluoro-2,4-dinitrobenzene, which we call FDNB. Okay, which we call FDNG, and we can use densyl chloride also. We can use dipsyl chloride also. Okay, here spelling is written wrong. Dipsyl, it is just a minute. I write. It's a dipsyl chloride. Okay, it's a dipsyl chloride. Okay, so we can use these three for the N-terminal amino acids. What happened? These molecule compound binds to our N-terminal amino acid. It means the amino acid like 1, 2, and 3. The amino acid which is on the N-terminal get conjugated with these compound dipsyl, densyl, or FDNB. Okay, to FDNB was reagent developed by Sanger. So many call this reagent as Sanger reagent also. What they call Sanger reagent. Okay, you can also do the C terminal analysis. Okay, how you can do the C terminal analysis? The carboxypeptidase. How? By carboxypeptidase we can do. It will remove the ex. It is what exopeptidase. It is what exopeptidase. This is the cut the C terminal amino acid. Okay. So how it is done? Let us see. I'll zoom the diagram. Just a minute. I think we need to do that only. Okay. So here you can see one picture. This is a polypeptide chain. Here we have done the N-terminal amino acids to know which amino acid on the N-terminal. At least we can know the two sequences like first and last two amino acid we can know. Okay, to know this we come to know about N-terminal amino acid the FDNB. It get conjugated with our N-terminal amino acid. Then we remove this amino acid. What we do? Then we remove this amino acid and the leftover chain goes aside. Okay, 6-HCl, we remove this amino acid by the acid hydrolysis again. Okay, so this derivative is again passed through HPLC. This derivative is again passed through HPLC. Okay, to know that what is the amino acid we had. Actually, we already had the standardization in our HPLC. Standardization. Okay, we already have the standardization in our HPLC. Okay, all amino acid derivative standardization we already have. So HPLC identify our amino acid by standard peak, so we can easily know about that. Okay, and the next step come for Edman degradation method. Actually, what is Edman degradation method? Let me tell you. In the there are two process. Basically, there are two sequencing process. One is by Edman degradation method, and another by MS spectroscopy. Sorry, mass spectroscopy. Okay, just a minute. Mass spectroscopy. Okay, so this we are studying Edman degradation method. Okay, but there is one limitation of Edman's degradation method that it can sequence only the polypeptide. It can only sequence the polypeptide which is up to which is up to 50 amino acid. Like it can sequence only that polypeptide which is made up of 50 amino acid maximum. Beyond that, it can sequence, but there will be loss of sequence. Like efficacy will not be there. Okay, like efficacy is not, then it will not give proper sequence. Okay, so Edman's degradation limitation is only this, that it can sequence only 50 amino acid. But we all know a protein is not just made up of 50 amino acid. It made up of more than 50. You can say more than 100, more than 200. To do the sequencing, what we need to do, we use some protein, we use some protease chemical. What we use? We use some protease chemical. We use some protease chemical. What this protease chemical actually do? Actually, suppose this is a polypeptide chain. This is made up of 200 amino acid. Okay. There are two protease chemical. Okay. Which are two protease chemical? One you can say exoprotease. Okay. Another you can say endoprotease. Okay. Exoprotease cuts where? At the N terminal and C terminal. Okay. At the exit side. It will cut at the end side. Okay. It will cut at the end side. And endoprotease. This one. Endoprotease. Endoprotease will cut at the middle of the chain. It will cut here. Our exoprotease will cut here. Okay. Exoprotease, I will indicate with this red one. It will cut over here. Endoprotease will cut in the middle of the polypeptide chain. Okay. So what we use? We'll use the endoprotease and exoprotease both protein our chain. Okay. Like I've said, it is 200. When we use the endoprotease, okay, endoprotease do what? It cut our chain in the middle. Okay. Then we get different size chain. Then we got different size chain. Okay, just a minute. Yeah, this you can see, like I have cut the chain, then we get two bands. Okay, we got the two chain of polypeptide chain. It is 100 maybe, then again we can cut with endopeptide, then it will again come this way. Just a minute, I'll show you once more time. Now see, I have cut one chain, we got another chain, we got. Okay, again if I cut this, again if I cut this, then again one two chain we got, then we got chain. Okay, so this way we got a short polypeptide chain which we can sequence. Okay, so now we have come for the sequencing. For the sequencing we are using the Edman degradation method. Okay, for sequencing, let us come here. For sequencing, peptide sequencing was done by Edman degradation method. Okay, we come by Edman degradation method. He used the Edman reagent. This is called the Edman reagent. Okay, what reagent is called? Edman reagent, which is called phenyl isothiocyanate. Okay, what it is called? Phenyl isothiocyanate. Like in the last one, I have told you that when we cut our chain, like we got the four chain in the last one. Okay, so now here is N terminal, C terminal, N terminal, C terminal, N terminal, C terminal, N terminal and C terminal. A four chain will be having. Okay, but what do Edman reagent do? Phenyl isothiocyanate do? Phenyl isothiocyanate bind to N terminal amino acid. What phenyl isothiocyanate do? Bind to N terminal amino acid. Okay, so what we do, one by one we do the sequencing for each chain. First, second chain, third chain, fourth chain. Okay, one by one we do the sequencing. How we do? We adhere one chain C terminal to one nitrocellulose membrane. or we can say one, there is some apparatus. Just a minute. Okay, C terminal. Just a minute. C terminal we fixed at one end. Okay. Then we give phenyl isothiionate. Okay. Phenyl isothiionate bind to what? Phenyl isothiionate bind to N terminal amino acid. Okay. It bind to N terminal amino acid. As soon as it bind to N terminal amino acid, in which condition it bind to N terminal amino acid? In the alkyl condition. It bind in alkali condition. As you can see, here is the diagram. You can clearly see. If not clearly, I am really sorry. You can increase your resolution. Okay. So here you can see bind. So this is the diagram which is formed. Okay. Phenyl isothianate bind with the first and terminal amino acid. Then we do the cleavage. Okay. Then we'll cut this N terminal amino acid by trifluoroacetic acid, TFA. Trifluoroacetic acid cut the derivative, okay, which is called thiazolinone derivative, which is called thiazolinone derivative, okay, which is called thiazolinone derivative, okay. So this derivative we got, okay. We don't know still which amino acid it is. We just know we just use phenyl thiocyanate, which bind to some kind of amino acid from the N terminal, and we bind this C terminal to one end. Okay. Now this is the actual thing happening. You can see here. This is the actual thing happening. Okay. So the amino acid, you can see this is a first amino acid, this is a second amino acid, okay. So this is phenyl isothiocyanate, it bind to this, okay, coupling reaction take place. After coupling, cleaving reaction take place. Then first amino ATZ amino acid, okay, adenosine amino acid derivative comes out with the first amino acid. Now second amino acid and the long chain is still there, okay. PTH amino acid, which is the derivative of ATH, this conversion reaction we used to do. And this derivative is again what we is done through HPLC analysis. Is done through HPLC analysis. Okay. So this cycle we used to run again and again. This cycle we used to run again and again. Like first amino acid is removed, R1 amino acid is removed. Okay. Now we again do the same thing with the second one, second amino acid, we again bind with it and we again get the sequence. This way we get the full polypeptide chain sequence. Okay, by this we get the full polypeptide chain sequence. Okay, but there is still one question. Suppose this we have taken primary structure. This we have taken just a primary structure. Now you can ask one more question. Suppose there are more than one polypeptide chain joined together. We know that it is joined together by disulfide bond, disulfide bond. So how we do the sequencing? For this thing, we need to come to the first step again. Okay, to know this thing. During the acid N terminal analysis, just a minute. Okay, during the N-terminal analysis, we are getting our N-terminal analysis, the N amino acid from the polypeptide chain. Suppose if the like we know that polypeptide chain has N-terminal, C-terminal. Suppose if there are two chain, sorry, if there are two chain, so there we will be getting two derivatives. It's possible now. We will be getting two derivative of FD and B. Okay, we getting two amino acids. So why this we come to know that there are two polypeptide chain. Okay, so we'll what do in the Edman degradation, but before cutting them, before cutting them from middle, okay, what we'll do? Just a minute. Okay, before the cutting them from the middle, we'll do, reduce them. We what do? We do the reducing step. We do the reducing step by beta mercaptanol, beta mercaptanol, okay, or DTT. Dithiothiol. Okay, we can reduce this and broke the disulfide bond. Then we can get the two separate chain. Then again we can separate that two chains, and then we can broken down them into their cleavage and just find the single polypeptide chain sequence. Okay, so this is all about the protein sequencing. Is I hope you all have liked the video. If you did not understand anything, just a minute. Okay. If you did not understand, if you didn't understand anything or had doubt, what you can do, you can comment. I'll try to reply each and every one if possible. So this is all about protein sequencing. I tried to explain you within a very short period of time. Like I had many doubts when I was preparing, learning about the protein sequencing, but now my many doubts are clear. So I hope if I find any doubts in further, it will get clear, and I'll get support from you all. So if you had any doubt, you can comment and subscribe the channel, and you can comment about like next which topic you want. This is the very short time I tried to explain you protein sequencing, and thank you so much for this video.